Hello today. My name is Filippo Petti. I thank the team at the Alliance for Regenerative Medicine for inviting us to participate in the Cell and Gene meeting on the MED for 2021. I'll be walking you through our Celyad Oncology story. I will be making some forward-looking statements, and I'll refer you to our publicly filed information for additional context into the company. So Celyad Oncology, we are really redefining the CAR T development pipeline. We are based and focused on allogeneic CAR Ts underpinned by two unique non-gene edited platforms. Both platforms use our all-in-one vector approach, which helps to streamline our design and development of CAR T, next generation CAR T therapies. Our first non-gene edited technology, TIM, is embedded in our CYAD 101 candidate for metastatic colorectal cancer. And we also have an shRNA platform, which is incorporated in our CYAD 211 program for the treatment of relapsed refractory multiple myeloma. We have been pioneering NKG2D CAR Ts over the last few years, really focused on both solid tumors and hematological malignancies. We really believe that NKG2D offers a novel approach for CAR T development, given the broad expression of the ligands that the receptor targets, and helps us think about driving towards a pipeline and a program strategy. We have delivered, and one of the only companies in the CAR T space, initial clinical data from a solid tumor indication from our CYAD 101 program. And we have recently announced a clinical trial collaboration in solid tumors to evaluate the CYAD 101 CAR T with Keytruda from Merck in the treatment of metastatic colorectal cancer patients. And the goal is to begin that study in the course of 2021. And really underpinned, all of this is possible through our robust intellectual property, both for US patents associated with allogeneic CAR Ts, as well as NKG2D receptor-based cell therapies. Just quickly highlighting our pipeline, as I said earlier, we are building a differentiated pipeline across allogeneic CAR Ts. We do also have an autologous program, CYAD 02, for the treatment of relapsed refractory AML and MDS. Currently, we sit with 3 clinical based stage programs, our CYAD 101 for metastatic colorectal cancer, which is an NKG2D receptor-based technology. The CYAD 211 program, which targets BCMA for the treatment of relapsed refractory multiple myeloma, which just recently entered the clinical stage phase 1. And our CYAD 02 program, which is also in phase 1 dose escalation for the treatment of relapsed refractory AML and MDS. A little bit of background on CYAD 101. This slide really captures where we've been focusing our efforts. Metastatic colorectal cancer is very much a deadly disease, unmet medical need. As you can see, it's the 3rd most diagnosed cancer worldwide. And specifically within the metastatic colorectal cancer patient population, there's 2 subtypes that seem to be a major focus. The MSI high patient populations which can receive checkpoint inhibitors, such as Keytruda and other PD-1 therapies. And then the majority of patients, are 95% of the patient population, are MSS patients or microsatellite stable. Here, rarely do immunotherapies work. There's really an unmet medical need in this patient population. And just on the right-hand side, just the key numbers that we focus on in terms of, for solid tumors, the ability to move progression-free survival and improve overall survival is a key focus of the organization. Additional background on 101. So 101, as I said earlier, is an NKG2D based CAR. It incorporates the TIM technology. It also incorporates a selection marker, allowing us to positively select for the allogeneic 101 cells that we produce. This is, again, based on our all-in-one vector approach. As you can see on the bottom of the right side of the schematic, everything is incorporated in a single gene construct. Allows us to have a single transduction and minimize the number of gene modifications that we need for development and manufacturing of the program. This, as I said earlier, CD3 zeta inhibits the TIM technology inhibits the CD3 zeta component of the TCR complex and helps us to reduce the potential for graft versus host disease. And the AlloShrink phase 1 trial kicked off in the end of 2018. On this next slide is some initial data from our dose escalation phase 1 trial. We evaluated the 101 program following FOLFOX chemotherapy. 3 infusions of the cell therapy, 2 days after the infusion of the chemotherapy, across 15 patients at 3 different doses, 100 million, 300, and 1 billion cells infused 3 different times. As you can see on the left-hand side, prior metastatic regimens and backgrounds in terms of driver mutations for these patient populations that you would typically see. You can think about this as a third, fourth line patient population. And the high level thoughts here is that we've seen favorable tolerability profile from the cell therapy, no dose limiting toxicities, no graft versus host disease from the dose escalation. Encouragingly, we've also seen some clinical benefit. We've seen 2 patients who had partial response and a few patients who had durable stable disease as well. Overall, the disease control rate was 73%. Our median PFS, or progression-free survival, for the 15 patients was 3.9. And the median overall survival, we recently reported as 10.6 months. And this compares to about 2 months and 8 months plus for the those metrics for agents that are currently approved for this patient population. And importantly, we've seen tumor burden decrease across 8 out of the 15 patients treated to date. So next steps for the program is to move into the expansion cohort of the study. And this slide just quickly captures the background of the expansion cohort. We will be evaluating CYAD 101 following FOLFIRI preconditioning chemotherapy. We'll be examining the 1 billion dose level per 3 infusions, as you can see on the bottom schematic. And really, this will allow us to enroll patients who have recently failed FOLFIRI and allows us to really tease out the benefit that the cell therapy may provide for these patients. The plan is to evaluate up to 34 patients in this dose expansion cohort, and preliminary data is expected by the end of the first half of 2021. Just quickly on the 101 CYAD, the CYAD 101 program with respect to our clinical trial collaboration with Merck and the Keytruda Keynote B79 trial. We announced in September of last year this clinical trial, which will evaluate Merck's Keytruda following CYAD 101 following preconditioning chemotherapy, specifically in microsatellite stable patients. The trial is expected to begin in the first half of 2021. And on the right-hand slide, we highlight the complementary mechanism of actions that we believe both NKG2D and anti-PD-1 allow for us to evaluate within the Keynote B79 study. And really the emphasis is the ability for NKG2D to resculpt the tumor microenvironment, to unleash the patient's immune system, to then be further enhanced perhaps with treatment with an anti-PD-1 therapy. Quickly turning over to the 211 program, our program for BCMA, our first allogeneic shRNA-based therapy. Here's a quick slide in terms of how we think about the universe. We are focused on a hairpin, simply put, a knockdown approach versus a knockout approach, as to many of our peers that who are using that in the context of allogeneic CAR-Ts. And some of the benefits of using the shRNA approach is listed on the left-hand slide. Succinctly speaking, there's no gene modification. We're able to mediate the gene knockdown and can titer up and down the amount that we want to regulate the gene expression. We can do this through a multiplex. We have shown data that we can knock down 4 genes simultaneously, which gives us certainly the opportunity to really define the phenotype of the CAR-Ts that we're looking to produce. It again leverages our all-in-one vector approach, allows us to also use a single-step enrichment process similar to what we've incorporated with our 101 program as well. And data that we have from in vivo studies shows that a knockdown approach using shRNA is comparable to a gene-edited approach in terms of reducing or protection from graft versus host disease, as well as we've seen some benefits in terms of perhaps seeing improved persistence using the knockdown shRNA approach versus a gene-editing CRISPR-Cas9 approach. So quickly, a little bit of background on 211. Again, you can see on the schematic on the right-hand side here, it is a BCMA CAR. It incorporates the shRNA, which allows us to knock down the CD3 zeta component of the TCR complex. And we also use a selection marker to help to enrich for the cells that we create, similar to what we discussed previously with the 101 candidate. Again, shRNA here interferes with the CD3 zeta complex, minimizes the expression of that of the TCR on the cell surface, and what we've seen to date in our preclinical models reduces the potential for graft versus host disease. Our Immunacy 1 phase 1 trial, which is evaluating CYAD 211 for the treatment of relapsed refractory multiple myeloma, kicked off at the end of 2020. This is just a little bit of background, as you can see, in terms of some of the data that went into the IND package, preclinical data for the 211 program. Again, on the left-hand slide, TCR function is knocked down. We help to validate this shRNA approach preclinically in terms of the TCR function, the ability for it to signal. In the middle is a gold standard in vivo graft versus host disease model, and you can see that we have protection in using allogeneic cells, the 211 cells in this gold standard model as compared to control, where you see the survival of those in vivo mice pass away over several weeks. And then lastly, the anti-tumor activity as we go out to 60 days is continued to be valid in terms of the CYAD 211 candidate in a KMS 11 xenograft model, which is a very standard model for multiple myeloma. So we can eliminate TCR function, we can have protection against graft versus host disease, and we see anti-tumor activity for the 211 program in vivo models. And so quickly, just a little bit of background on CYAD the CYAD 211 in Immunacy trial. We are going to evaluate the 211 following a single infusion of 211 after preconditioning chemotherapy with cyclophosphamide fludarabine. We'll be looking at 30 million cells, 100 million cells, and 300 million cells in our dose escalation program. Recruitment is ongoing in the first dose cohort, and we expect to announce initial proof of concept data for the shRNA technology program, the first 3 to 5 patients for the Immunacy 1 trial by the end of the first half of 2021. And then additional clinical activity data will be available by year-end 2021. Lastly, just to quickly reiterate, the beauty and the dynamic ability of the shRNA platform is for us to knock down multiple genes and proteins at the same time. And this is just a quick slide showing that in a single construct, we were able to knock down 4 genes simultaneously, things like beta-2M, which is a gene and protein of interest for the persistence of allogeneic cells, DGK, which is an internal kinase. Again, one of the differentiating parts of the shRNA is that we can target internal and then select for those cells that are being targeted for that knockdown. CD3 zeta, as we've shown earlier, a single knockdown for our 211 program, and then CD52, again, another protein of interest that is being evaluated by some of our peers in the context of increasing persistence of the allogeneic CAR T cells. So our next generation candidates will explore these multiple shRNA knockdown approaches. Quickly turning to our O2 program for autologous CAR T for the treatment of relapsed refractory AML and multiple myelodysplastic syndrome. O2 is our next generation NKG2D autologous program. It also incorporates some shRNA technology, which targets the NKG2D ligands MIC-A and MIC-B. In the first quarter of 2020, we initiated the phase 1 cycle 1 dose escalation trial, which will evaluate O2 following preconditioning for the treatment of AML and MDS. We're currently enrolling patients in the study, and we're evaluating 3 dose levels for the trial. The shRNA technology, and right, just a little bit of background here, it has shown to be able to knock down the expression of the MIC-A and MIC-B ligands on the CAR T cells that we generate, the autologous cells, and that allows us, we believe, to provide additional in vitro expression and expansion of the cells, and perhaps may have an impact based on our in vivo models on clinical activity. And just a little bit of data from our phase 1 trial to date. This was data that was presented at the ASH 2020 annual meeting, and it compares our CYAD 02 next generation candidate with our CYAD 01 candidate, and data that we've generated in phase 1 dose escalation trials. Based on the data you can see from O2 on top, we believe at similar dose levels that because of the incorporation of the MIC-A, MIC-B targeting shRNA, we're able to improve some of the anti-leukemic activity to date that we've seen, as well as perhaps the duration of those stable diseases that we've been able to, in patients that we've been able to enroll. When we compare to that to the O1 program, which again looked at similar doses following preconditioning chemotherapy of cyclophosphamide fludarabine, really the only change here is the shRNA technology, and really why we believe it could be leading to a differentiated profile for this NKG2D autologous program. So dose level 3 is ongoing, as I said. Additional safety and clinical activity data will be available during the first half of 2021. So just to quickly review our upcoming milestones for the first half of this year, we anticipate to report preliminary data from the expansion cohort of the AlloShrink trial that is currently on enrolling patients, and to initiate the phase 1b Keynote B79 trial with Keytruda in patients with metastatic microsatellite stable disease with metastatic colorectal cancer. The 211 program, as I highlighted earlier, dose enrollment is ongoing in the dose escalation trial. We plan to have initial data from the first cohorts during the first half of this year for the Immunocy phase 1 trial for relapsed refractory multiple myeloma. And at the end of the first half, we also anticipate some additional data from the CYAD O2 program, the cycle 1 trial specifically for the treatment of relapsed refractory AML. So a very data-rich calendar year for us, in part specifically for the first half of 2021. With that, I'd like to thank everyone for your attention, and again, appreciate the Alliance of Regenerative Medicine for inviting us to the Cell and Gene meeting on the Med for 2021. Thank you.